Hey everyone, welcome you all to Bioschool. This is Gayatri. Hope you all are fine and preparing well for your examination. In today's session, we will see regulation of gene expression in prokaryotes. This is the part 3 where we will focus on the response of Lac Operon to the different conditions of lactose and glucose concentration. In previous two videos, we have discussed positive and negative regulation of Lac Operon. You will get the link in the description box down below. So let's go ahead and know how the Lac Operon it responds to different conditions of lactose and glucose. First we will see in presence of lactose. If in the media lactose is present, so whether the Lac Operon it will turn on or turn off. We will see now. This one is the operon. Operon it consists of a group of structural genes. The structural genes are LAC-Z, LAC-Y and LAC-A. These three structural genes are controlled by a single promoter region. The LAC-Z it encodes an enzyme. That enzyme is known as the beta-galactosidase, whereas LAC-Y it encodes the enzyme that is formase, and LAC-A it encodes the enzyme beta-galactoside transacetylase. So when in the media lactose is present, then at that time beta-galactosidase enzyme it will cleaves the beta-1,4 glycosidic linkase which is present in between the glucose and galactose, and it will form the glucose molecules as well as the galactose molecule. At the same time, beta-galactosidase it also forms a beta-1,6 glycosidic bond in between the glucose and galactose. That compound which is formed is known as the allolactose. Okay. So if lactose is present, then it will convert it into allolactose. Allolactose it acts as an inducer and it induces the Lac Operon. That's why Lac Operon is known as the inducible operon. If the inducer is present, then this inducer it will bind to the repressor. Okay. And the conformation of repressor it will change, and the repressor now it cannot bind to the operator region. So operator region it is now free. Now RNA polymerase it can bind to the promoter region and it helps in the transcription process. So if the glucose is present, then transcription process it will be on. Okay, now we'll see in absence of the lactose. If lactose is absent, then there will be no production of allolactose. That is, the inducer is absent. So in absence of the inducer, the repressor protein, it will bind to the operator region. And as we know that promoter region and operator region, they are overlapping with each other. When the operator it binds with the repressor, then at that time RNA polymerase it cannot bind to the promoter region. So thereby the Lac Operon it will be turned off. Now we'll see in presence of low concentration of glucose. So if the glucose is present in the low concentration, the enzyme adenylyl cyclase it will be active and it convert the ATP to cyclic AMP. So this cyclic AMP it will bind to a protein. That protein is known as the catabolite activator protein, that is the CAP protein. So when the CAP protein it binds with the cyclic AMP, then they together it will bind to a site that is known as the catabolite activator protein site or CAP site, that is present just upstream to the promoter region, right? So when the CAP site it is bind with the CAP protein along with the cyclic AMP, CAP protein it cannot bind to the CAP site in absence of cyclic AMP. Okay. When in the media the glucose concentration will be low, then cyclic AMP produced. So when cyclic AMP is present, then the CAP protein it will get activated and it will bind to the CAP site. As the CAP protein bind to the CAP site, then it will enhance the binding of RNA polymerase to the promoter region. That means the RNA polymerase now can bind efficiently to the promoter region and it enhances the rate of transcription. At the transcription rate it will be high. Otherwise In absence of the cap protein, RNA polymerase, it cannot bind efficiently to the promoter region because the promoter of Lac Operon, it is a weak promoter. Now we'll see in presence of high concentration of glucose. If the glucose concentration is high, then it will inhibit the adenylyl cyclase. So there will be no production of cyclic AMP. As the cyclic AMP, it is absent. So what do you think? Cap protein, it will be active or it will be inactive? It will be inactive because for its activation, it needs the cyclic AMP. As the cap protein, it is inactive, so it cannot bind to the cap site. Now the RNA polymerase, it will bind to the promoter region, but the promoter is weak promoter, so it cannot bind efficiently and it will make very few transcript, that is results in the low transcription. Okay, and the glucose concentration, it regulates positively, and the cap protein is positively regulate the Lac Operon. If cap protein is active, then it will enhance the rate of transcription. If cap protein is inactive, then it will lower the rate of transcription. Now we'll see the another condition where glucose is present but lactose is absent. The simple formula is that if in the media lactose is absent, then why the genes which are helping in the catabolism of lactose, it will active? It will, of course, it will inactive and there will be no transcription, right? If lactose is absent, that means there will be no allolactose. In absence of inducer, the repressor protein, it will bind to the operator region. So as the operator and promoter region are overlapping with each other, so now RNA polymerase, it cannot bind to the promoter region, thereby there will be no transcription. Okay. And in the other condition, if you will look, the glucose is present. Though the glucose is present, but lactose is absent. That's why the Lac Operon, it will be turned off and there will be no transcription. Let's see the other condition where the glucose and lactose both are present. If lactose is present, then it will convert it into allolactose. Allolactose, it acts as an inducer and it will bind to the repressor protein. And the repressor, now it will change the conformation and it cannot bind to the operator region. Now the RNA polymerase, it will bind to the promoter region and it will help in the process of transcription. Now we'll see what is the effect of glucose. If the glucose is present, then the cyclic AMP, it cannot produce because it will inhibit the enzyme adenylyl cyclase. In absence of the cyclic AMP, cap protein, it is inactive. It cannot bind to the cap site. That means here, though the transcription, it is on, but the rate of transcription is low because the promoter of Lac Operon, it is a weak promoter, right? So in presence of glucose and lactose, the Lac Operon, it will be turned on, but the rate of transcription, it will be low. Other condition, glucose is absent and lactose is also absent. So simple, if the lactose is absent in the medium, then there will be no transcription. Okay, if lactose is absent, then the repressor protein, it will bind to the operator and thereby it will prevent the binding of RNA polymerase to the promoter and inhibit the transcription. Or there will be no transcription. Another condition, glucose is absent and lactose is present. Then what will happen? Lactose is present. That means it will convert it into allolactose, which will act as an inducer and it will bind to the repressor. So the repressor, it cannot bind to the operator. Now RNA polymerase, it will bind to the promoter, thereby transcription process, it will be on. Now we'll see the effect of glucose. Glucose is absent in the media or glucose is present at a very low concentration. If the glucose is absent, then it cannot inhibit the adenylyl cyclase, thereby it will produce the cyclic AMP. So when the cyclic AMP present, then this cyclic AMP now can bind with the cap protein. So when the cap protein, it binds with the cyclic AMP, the cap protein, it will get activated. Okay, now the cap protein, it can bind to the cap site. So when the cap site, it is bind with the cap protein, then it enhances the binding of RNA polymerase to promoter region. RNA polymerase, it will bind effectively to the promoter region. So here the rate of transcription, it will increase, or the rate of transcription will be strong. More transcript will be produced, right? So the transcription, it will be strong. Okay, now we'll have a look on the summary of Lac Operon. At which condition Lac Operon will be turned on or turned off? So in this condition, first we will look into the lactose concentration. If the lactose is absent in the medium, then the level of transcription, it will be off, or there will be no transcription. If the lactose is absent, then no need to look at the concentration of the glucose. It will direct, you can write that there will be no transcription. And similarly here the lactose is absent. Lactose absent means there will be no transcription. Here the lactose is present, that means the transcription, it will be on. But now we'll look into the concentration of glucose to know whether the transcription rate, it will be high or low. So in this condition, glucose concentration is high. If the glucose concentration is high, then cyclic AMP concentration, it will be low, then it cannot bind with the cap protein. So the cap protein is inactive. Okay, so there will be no production of cap-cam complex. That's why if there is the no activation of cap protein, so it cannot effectively bind the RNA polymerase to promoter. RNA polymerase now binds with the promoter very weakly. It will result in the very low level of transcription. Okay, in the last condition, lactose is present, but see here the glucose is low. If the glucose concentration low, then the cyclic AMP concentration high. If the cyclic AMP concentration high, then it will bind to the cap protein, and now the cap protein, it is active and it will form the cap-cam complex, and this cap-cam complex, it will enhance the binding of RNA polymerase to the promoter region. So now the RNA polymerase, it binds efficiently and it will result in the high level of transcription. Okay. So in the positive regulation, you have to remember this point, that when the glucose concentration high, then the cyclic AMP concentration, it will be low. If the cyclic AMP concentration low, then it cannot form the cap and cam complex. Then it cannot form a complex with cap protein. If the glucose concentration low, then the cyclic AMP concentration, it will be high. Then it will form a complex with the cap protein, and this active cap protein, it will enhance the rate of transcription, or the rate of transcription, it will be high. Now we'll see the previous year CSIR question. The Lac Operon in E. coli is controlled by both Lac Repression and Catabolite activation protein and the cap. Lac Repression, it is negatively regulate, and Catabolite activation protein, it positively regulate the Lac Operon. In an in vivo experiment with Lac Operon, the following observations were made. Let's see the options. Cyclic AMP level are high. Cyclic AMP level high means the glucose level is low. Okay, or glucose is absent. Repression is bound with allolactose. Here the allolactose is present, inducer is present. That means lactose, it is present in the media. If the lactose is present, then beta galactosidase, it form allolactose. Lactose is present. Cap is interacting with RNA polymerase. Cyclic AMP level high. That means cyclic AMP, it will bind to the cap protein and it will interact with the RNA polymerase. That it shows the absence of glucose or low concentration of glucose. Which one of the following question is most appropriate based on the above observation? So first is, glucose and lactose are present? No. Glucose is present. Glucose is present and lactose is absent? No, is the opposite. Both are absent. It's also wrong. Glucose is absent. Yes, when the cyclic AMP concentration are high, then glucose concentration is very low or it is absent. Okay, so glucose is absent and lactose is present. This question, it carries 4 marks. Thank you all for joining me today. Hope now you have a clear idea about Lac Operon, and how the Lac Operon it is regulated in different conditions of presence and absence of lactose and glucose. Still, if you have any confusion regarding the Lac Operon, then please put a comment in comment section down below. I will get back to you as soon as possible. In coming session, we will look into how merozygotes responds to the Lac Operon. That is very important. So if you have not subscribed my channel, then please do subscribe and press the bell icon so that you will get notified. Bye bye. See you in my next video.